We're quite pleased to have the opportunity to speak with you today. So our keynote speaker will be Dr. Charles Sawyers. And I think, Alan, we can go to the next slide. So I wanted to give you a very brief overview of the drug resistance and sensitivity network that was established as part of one of the Moonshot Blue Ribbon Panel initiatives to overcome drug resistance. The primary directive was to develop ways to overcome cancer's resistance to therapy, to launch a large interdisciplinary initiative, and to evaluate new therapies that prevent or overcome a tumor's ability to resist or become non-responsive to therapy. To that end, we developed the Drug Resistance and Sensitivity Network, sometimes referred to as DRSN. It is made up of 5 primary U54 grants, and in the center is both our coordinating committee and our is the coordinating committee. So it also uses a number of NCI resources. Next slide. So the goals I'll talk about the resources in just a minute. The goals are to create a drug resistance and sensitivity network, to focus on these issues broadly, and to have it be part of an integrated NCI precision medicine initiative. The objectives will focus on new models and diagnostic techniques that use human tumor samples. Investigators have components of their research involve druggable targets and the use of an iterative approach between the bench and the bedside. While these studies often use NCI IND agents, they are able to use other agents. Listed on the right-hand side of the slide is each of the U54s, their location and their focus at Mayo and University of Minnesota. Let me try that again in English. University of Minnesota is the myeloma resistance group that is working on IMiDs and proteasome inhibitors. The U54 at MGH and Dana-Farber is working on colorectal cancer, melanoma, and lung cancer resistance, looking at MAP, RTK, and immune checkpoint inhibitors. The U54 at MSKCC, which you'll hear a bit more about today, and the Hutch, focuses primarily in prostate cancer, and I'll leave that to Dr. Sawyers to discuss today. The U54 at the Oregon Health Sciences University works on acute myelogenous leukemia and intrinsic and extrinsic targets for combination therapies. And finally, the U54 at UCSF in Stanford evaluates lung cancer and mechanisms of resistance there. We have a coordinating center, which is run under the U24 grant at Roswell Park. Next slide. This is kind of the big picture. It's a busy big picture, but I think that's a good thing. And it really shows how each of the U54s and all of our grantees are working either together with each other or with any other NCI networks listed in the lower left-hand corner of the slide, or other NCI resources listed in the lower right-hand corner of the slide. So each grant also has attached to it a number of supplements that work for a year or maybe a little bit longer to do coordinated research with them. So go to the next slide, please. So I'm going to introduce Dr. David Goodrich, who is a professor of oncology at Roswell Park. His area of expertise in the department he works in is molecular pharmacology and cancer therapeutics, and he's also part of the graduate program and cancer genetics program as part of their CCSG grant. So with that, we'll go to Dr. Goodrich.
Thank you, Dr. Ivy. Welcome everybody to this month's Moonshot seminar. It's my honor to introduce today's speaker, Dr. Charles Sawyers. I look at the participant list and I see 418 participants. That's probably the biggest Zoom call I've ever been on. So I think it means that most of the people are probably very aware of Charles's work and his impact on science. So I will try to be brief. Dr. Sawyers earned his bachelor's degree at Princeton University, his MD at Johns Hopkins. He did residency at UCSF, then fellowships at UCLA, where he began his academic career. as a professor. He now serves as chair of the Human Oncology and Pathogenesis program at Memorial Sloan Kettering Cancer Center, and he's an investigator in the Howard Hughes Medical Institute. I think all of us who go into cancer biology have a dream that one day our work in the lab will eventually improve patient outcomes. And it's rare when one of us is able to accomplish that. It is rarer still when a scientist accomplishes that multiple times. And I think it's fair to say that Dr. Sawyers is one of those scientists. In 2009, he earned the Lasker DeBakey Clinical Medical Research Award for his participation in the development and discovery of ABL kinase inhibitors imatinib and dasatinib that are used to treat chronic myeloid leukemia. Simultaneously, he was working on improving anti-androgen therapies for the treatment of prostate cancer. And this work ultimately led to the co-discovery and development of enzalutamide. Enzalutamide was FDA approved in 2012, and it's now an important component of standard of care for most prostate cancer patients. In addition to this clinical impact, Dr. Sawyers' lab has made several important fundamental discoveries relevant to the mechanisms driving prostate cancer, such as those involving ETS factors, transcription factor FOXA1, PA3 kinase signaling, and others. And his lab continues to focus on mechanisms of resistance to cancer therapy. Dr. Sawyers has earned a number of additional honors for his scientific accomplishments. I'm just going to mention a few. He's a member of the National Academy, a member of the Institute of Medicine. He won the American Cancer Society Medal of Honor for Clinical Research just a few years ago. And he has served as past president of AACR and is a member of the National Cancer Advisory Board. I had the privilege of begin collaborating with Charles's lab about 4 to 5 years ago when we recognized our shared scientific interests in prostate cancer lineage plasticity as a mechanism of resistance to anti-androgen therapy. And today, Dr. Sawyers is going to share some of his recent discoveries in this area. And the title of his talk is Lineage Plasticity and Drug Cancer Resistance. Dr. Sawyers?
David, thank you for that really kind introduction. It's great that I also get to describe our collaborative work in this lecture. I also want to thank Percy and the NCI leadership for the honor of asking me to give this kickoff Moonshot seminar. I had the privilege of participating in the Blue Ribbon panel back, now 4+ years ago. And on that, through that exercise, I had the opportunity to meet the then Vice President Biden on multiple occasions. And when I got the invitation to give this lecture, it was not lost on me that the date was going to be roughly 2 weeks after the election. And of course, I'm actually quite happy that I get to hopefully see the former vice president now as the president-elect and interact on this continuing initiative. So let's get started. Here's my disclosure slide. David mentioned enzalutamide. That word will appear in my lecture, but I'm not going to make any claims about its activity. And again, as sort of a nod to the whole rationale behind the Moonshot Initiative, I want to start with a patient. This is a man who was seen at Sloan Kettering a couple of years ago by my colleague Yu Chen, and the pathology was worked up by Anu Gopalan. And the point I want to make is that this patient, like all patients, has a multi-quadrant biopsy. And this biopsy shows adenocarcinoma histology, whereas this biopsy shows a small cell histology. This is quite unusual as a presentation of prostate cancer, but it makes a very important point that will be a theme throughout my talk. And that point is that when you do molecular sequencing of these independently obtained biopsies, you see that they have a common origin. You can see that for the various alterations in black. And the small cell one has new alterations present in red. And the two that are most relevant for today are the P53 mutation that's present in both biopsies and the retinoblastoma gene deletion that's present in the small cell biopsy. So with that as sort of a foray into the question from a clinical anecdote, let's get started. So I just want to show one or two introductory slides to orient everyone into prostate cancer. So the disease is a luminal adenocarcinoma. The luminal cell is present in the normal gland. It's an androgen receptor expressing cell. These cells are dependent upon the androgen receptor for their maintenance and survival. And the cancers have been treated when they metastasize for decades based on that principle. The androgen receptor is required for the sustenance of these tumor cells. And that has been the basis for all these different versions of anti-androgen therapy, which I'm not going to get into in any great detail. But I want to make sure you're clear that the reason these drugs work is because it's a lineage survival factor in the tumor cells. It's not a driver oncogene that's being targeted. So I'm going to actually, my lecture is going to consist of sort of three components. I'm going to start with some analysis of the normal prostate using single cell approach. The reason to do this is because in order to understand this problem, particularly the plasticity problem, we need to know who the players are, and the power of single cell analysis has really opened the door for us to gain new insights at a level that I couldn't have imagined even just a few years ago. The second part will then flip back to that patient vignette, and I will go through examples of two model systems in which we modeled that, much of which has been done in collaboration with Dr. Goodrich. And then I also want to point out recent findings looking at human tissue as part of the U54 in collaboration with colleagues at the Fred Hutch. So let me get started with the single cell analysis. So this was published earlier this year. It's the work of Wouter Karthaus in my lab in collaboration with Aviv Regev's group at the Broad Institute and Donna Pear's group here at MSK. And all I want you to take away from this first slide is that there's greater complexity, not surprisingly, than was present on that cartoon I showed you earlier where there were just basal cells and luminal cells. There is one large cloud of basal cells. There's a large cloud of luminal cells, but it turns out there's at least two other smaller populations of luminal cells that have really interesting biology. I won't have time to go through all of it, but I'll touch on the main points and refer you to the paper. And I won't have any time to tell you about another entirely separate component of this it's also fascinating, and that is the microenvironment. So there's two different mesenchymal populations outlined here, and there's other smooth muscle populations. And if you read the paper, you'll see that each of these expresses different peptide growth hormones that will be familiar to you because they are well known to stimulate different epithelial subsets of cells. And so the crosstalk that's going on in the normal gland is revealed through this lens at a level that we hadn't previously appreciated. But to dig deeper into the luminal cells, if you just focus on analysis of the luminal cells and plot a new t-SNE analysis, you now see these three different subgroups in greater clarity. You can also see some of the markers that define them. This large cloud is what I like to refer to as the sort of terminally differentiated secretory luminal cells. They're marked by genes such as NKX3.1 and probasin, whereas the luminal 2 cells represent a smaller population with stem-like gene expression profiles such as SCA1, Ly6A, PSCA, and so forth. If you use immunofluorescence or immunohistochemistry to now ask, anatomically, where are these cells? The answer is the L2 or stem-like marker cells are present in the proximal part of the gland, whereas the L1 cells tend to dominate in the distal part of the gland, the more predominant cells. And I hope you can see this with some clarity, but I'm drawing my pointer to a sharp demarcation between the L2 cells, the stem-like cells in red expressing TROP2, and then the L1 cells in this cyan color. So, and this is occurring right here. And this is, I think, indicative of some kind of instructive signal during development that's defining the eventual emergence of these populations, which it will be, very interesting to learn more about. I want to also, acknowledge that other groups have also recently published on the topic of annotating the mouse prostate or human prostate at a single cell level. And friend and colleague Michael Shen shared this slide with me who's at Columbia. This paper came out recently in eLife. And although there are different nomenclature describing the different cell types, the theme is very consistent. There's a stem-like population that's proximally localized and a more secretory population that's distally localized. This also makes the point that there are different lobes of the mouse prostate, and there are some differences amongst the cells depending on which lobe you analyze. So that's sort of the anatomy at a single cell level. I now want to tell you about a couple of experiments in Wouter's paper that address some of the biology of these cells. And the way we addressed this initially was to put the mice through a castration regeneration cycle. And this is a well-known phenomenon that the prostate will regress following castration. It takes about 28 days. But remarkably, if you give testosterone back to the mouse, you can regenerate a fully normal appearing prostate in 28 days. And you can get a sense of that from this panel on the far left, from an intact mouse, and this on the far right from a regenerated prostate. And you can see how the lumen, restores itself to normal. The main point I want you to take away from this time course is the Ki67 staining in red. And at baseline, there's just a few pink or red cells. But what I hope you can see is that at day 3 after regeneration, it looks like almost the entire gland is lighting up with the Ki67 marker. This was a little surprising to us because the general thinking of prostate regeneration has been that there perhaps is a stem-like cell that's responsible for repopulating the entire gland. And as I showed you, there is this L2 population that's a minority population that was a sort of very strong candidate for that cell. But it'd be hard to imagine that cell could proliferate this quickly to give that kind of signal. So we looked at this in more detail using the single cell RNA-seq analysis. And again, I don't have time to go into all the details of these fate graphs, but what we've done is plotted cell signatures across this time course. And what you'll just have to look to the paper for the details, but what's happening is an intact mouse cells are over here, and during castration, they go in this direction, and then during regeneration, they come back. And if you look at the different L1, L2, and L3 cells, the L1 cells undergo remarkable transcriptomic changes during this cycle. L2 cells less so, and L3 cells, which I didn't touch on, are not really changing, even though they express the androgen receptor. This now also allowed us to look at the fraction of Ki67 positive cells as numerated by messenger RNA now from the single cell sequencing analysis across this time course and broken down by L1, L2, and L3. And what I want you to see here is that 2 days after androgen add back, you see that 68% of the L1 cells and 45% of the L2 cells are now in cycle. So it really is entirely consistent with what I showed you on that previous immunofluorescence of the gland. So this raised a question. Well, first of all, one more confirmation of that result. So I think this is going to be a little too faint for you to see, but this is 2 days after regeneration, and it sort of confirms what I showed you earlier. There's lots of Ki67 staining throughout the gland, but now I'm giving you the orientation of proximal versus distal, and the main reason I'm doing that is because you see a lot of this happening in the distal gland where the L1 cells reside, as well as where the L2 cells reside in the proximal region. So this set up an interesting question, which Wouter addressed through a lineage tracing experiment. So we took advantage of the fact that there's a Cre driver that's expressed specifically in luminal cells and not basal cells, as well as this confetti mouse in which different colors, fluorescent colors will be selectively activated by Cre, and you can use it to mark the relative growth or progression of differently marked cells. So the setup of the experiment is to put the mice through castration, fully regress the gland, give tamoxifen to activate the Cre, wait a week to then confirm that you've deleted and activated a color in different cells, and then give testosterone and see what happens. And Wouter, in his humorous way, depicted the two different possibilities. One would be that every cell is contributing to the regenerated gland, and the other, more consistent with a stem cell type model, is that there would be a few clones that dominate. And the answer is shown here. This first panel on the upper left shows just 7 days after tamoxifen that we are getting single cells marking with different fluorescent colors. And then 4 weeks later, what I want you to take away from this is that this pattern looks pretty similar, except now the clusters are somewhat larger, and here's a higher power view. And we carefully annotated the number of cells per cluster across hundreds of these, and that's all described in this plot here. And the conclusion is that these cells undergo 2, maybe 3 doublings over this period of time. And so it really supports, if you will, the communist model. Now, another aspect of the functional characterization was to use cell surface markers that allow us to selectively isolate L1 versus L2 cells, and those are listed here, and then put those cells into organoid culture as single cells and measure their cloning efficiency, which means their ability to form an organoid that has an appearance like this. And as expected from intact cells, from intact animals, the cloning efficiency of the terminally secretory cells is low. It's about 4%. Whereas the ones that are SCA1 positive, that minority population, it's about twice as high. So this fits a lot of prior data from us and others. But what caught our attention was the increase in cloning efficiency following castration. And furthermore, that increase went up even further during those first 2 to 3 days of androgen add back to now about 1 out of 5 cells. So there's a couple of sort of takeaways from this result. One is that these cells undergo a cell state change, at least in terms of repopulating a regenerative capacity induced by castration. We don't know the mechanism for this, but it's quite interesting, of course. But we should also be thinking about sort of the medical implications of this, because we give castration therapy so often to these patients. It if you will, could be a bit of a double-edged sword, where we're giving it for the right reasons of inhibiting or killing the growth of the androgen receptor dependent tumor cells, but we also are potentially activating a self-renewal program. So I show you this slide again to just now sort of signal to you that I'm going to shift from the single cell analysis of normal to the plasticity question that I introduced earlier. And the reason I pointed your attention to RB and P53 is based on the frequency of alteration of these two genes in genomic landscape analyses of prostate cancer. And it may seem surprising, but despite how famous these tumor suppressors are, they hadn't really attracted much attention in prostate cancer until recently. And that is primarily because the frequency of alteration in primary cancer, at least as defined by TCGA and confirmed by many other studies is quite low, 8% and 1% respectively. In contrast, in metastatic disease, roughly half the patients have a p53 alteration and about 20% have an RB alteration, and a consistent number of those have both. So with that as a backdrop, I want to share just the sort of one slide on two back-to-back papers that David's group and our group published a couple of years ago. The first one is the work of a former postdoc, Ping Mu, who's now at UT Southwestern running his own group. And the takeaway from this work is that if you take prostate cancer adenocarcinoma models, human models that are wild type for RB and p53, and then use various tricks like CRISPR or inducible shRNA knockdown and target RB and p53, you can get resistance to castration therapy or enzalutamide. But you have to target both. And what was sort of eye-catching about this result is that the reason they develop resistance is because they lose their luminal identity through a plasticity phenomenon that in this paper we showed was dependent in part on SOX2. And the reason that's fascinating is because until this, really the way we were thinking about resistance to anti-androgen therapy was sort of on this left-hand side, where adenocarcinomas relapse, they still look like adenocarcinomas, and in general, they have somehow figured out a way to restore androgen receptor target gene expression through these various mechanisms. And that in the companion paper led by David's group, he described 3 genetically engineered mouse models which contain either 1, 2, or 3 deletions of a cassette of tumor suppressor genes, PTEN alone, PTEN with RB, or the triple, which is PTEN, RB, and p53. And what was remarkable about this work is that in the constellation of the double deletion or the triple deletion, the mice would develop classic adenocarcinomas initially, but they would evolve to a neuroendocrine phenotype, similar to what I showed you in that patient example, but now in this case, evolving during disease progression in a mouse model that we can interrogate over time. And there's lots of details about the 2 models, which I refer you to the paper for more questions, but the TKO develops quite rapid onset disease. The DKO model takes longer, and in both models, particularly evident in the DKO, if you give castration therapy, you can accelerate the rate at which the neuroendocrine transition occurs. So what I want to now show you is unpublished work from our collaboration using our experience now with single cell analysis to do a time course across these mice. So here's the models and the different time courses. This work has been led by Samir Zaidi in my lab with Jimmy Chow in collaboration with Joe Chan and Donna Pear for the computational analysis. And I mentioned David and we've been working closely with Christine from his group. So I want to sort of hit just the high details, but here's a map of this plot based on genotype. So you can see that there's gray clouds here in the wild type that indicate that there's big mutation-specific gene expression clusters of cells, and I've made this a little easier for you to take away by showing it in this way, where I've defined them based on lineage expression profiles that define them. And also the color here is GFP, which is important because it allows us, because GFP is a marker that's also activated by the Cree driver, so we can trace which cells have the tumor suppressor deletion profile. So first of all, you can clearly see the neuroendocrine cells as a distinct cluster. You can also see that there's a large cluster of luminal epithelial cells here that are tumorigenic. I won't show you the data, but if you ask what type of luminal cells they are based on that L1, L2, L3 analysis, they look very much like the L2 stem-like cluster. The other point I want to make, and won't spend any time on, is there a cluster of green cells in this mesenchymal cloud that we believe are derived from the luminal epithelium, although it's difficult to absolutely prove that it's not related to some kind of leakiness of Cre in the stromal compartment. But I'll show you some computational evidence on this slide that may not be the case. So what I'm showing you now is just looking at the epithelial cells. And this is a phenograph that defines 11 different clusters. And sort of at the center here is the transformed luminal population. And what's gotten us excited are these trajectories that point to the neuroendocrine subpopulation, as well as two other subpopulations that we really had not even thought about in our minds. One is marked by a transcription factor known as PAU2F3, and the reason that's super exciting to us is because this is a transcription factor that defines a subset of small cell lung cancer. So a common theme here that may be relevant in the human disease. There's another cluster defined by a trefoil gene called TFF3, again, a master regulator transcription factor. We need to know a little bit more about this before we can make any definitive conclusions. This shows expression of, for example, AR target genes, and you can see that the luminal cells, as expected, are androgen receptor expressing. The neuroendocrine cells are not. Cell cycle is also quite interesting, perhaps not surprising. The neuroendocrine population has a large S-phase fraction, which is consistent with the clinical behavior of these types of patients at this stage of the disease. I also want to call your attention to a gene expression signature that really jumps out, and that's JAK-STAT activation and downstream interferon response genes. And you can see the levels are here. And if you also trace this, to the clusters 6 and 7, that's these clusters here, which we think are, if you will, the sort of the seed for the plasticity phenotype with these different trajectories. But I'll also point out that the neuroendocrine clusters defined here by sort of 10 and 11, we don't see that inflammatory signal anymore. And I'll come back to that. Another takeaway from the mice is that within the neuroendocrine cluster, there's more heterogeneity than might initially meet the eye. And so again, too much detail here to go into in great depth, but the main takeaways are there's a cluster that we call NEPC-A, which is defined by the sort of master regulator neuroendocrine transcription factor, ASCL1, and you can see that pattern here. Together with INSM1, well known in the small cell lung cancer field. But we also have this, what we call NEPC-P, which is the PAU2F, and we also, PAU2F3, that's marked here. And we also have this group here, which is chromogranin positive, as well as androgen receptor positive. And Pete Nelson's group at the Hutch has described this amphicrine type form of prostate cancer, which is sort of a dual identity between expressing both, luminal markers as well as neuroendocrine markers. I'm now going to show you just a little bit of analysis from human late-stage prostate cancer at a single cell level. So this is a small cohort of just 5 patients at this stage, and Samir has done this analysis together with our clinical colleagues in GU Oncology. And my main point in showing you this is that, RB and P53 are deleted at high frequency in these late stage samples. And we also see a similar level of heterogeneity. For example, we see what looks like the human analog of the ASCL1 phenotype. We also see an interesting variant in which they don't express chromogranin A, but they express chromogranin B. And we see poorly differentiated adenocarcinoma cells that express low levels of the PAU2F3 transcription factor, but and may or may not be the sort of human equivalent of that small cell variant. There's also further, evidence of the heterogeneity shown here. Again, expression of these, and now showing you some of the plasticity transcription factors such as SOX2, SOX9, and so forth that change across this continuum. So in addition to that small cell analysis, one of the great benefits of participating in the DRSN network and having our partners at Fred Hutch was the chance to work with Michael Hafner and Martine Roudier in pathology at Fred Hutch. And the group at the Hutch is well known for having initiated, a long time ago, a rapid autopsy program in prostate cancer. So they've collected a unique set of samples that are now annotated on a tissue microarray. So taking all the various markers that came out from the mouse profiling and all the sort of parallels with small cell lung cancer, we decided to interrogate this microarray with a large number of antibodies. I'm also listing, Anu Gopalan, who's our GU pathologist at MSK, as well as Natasha Reckman here at MSK, who's our lung pathologist, because in order to get this, to work efficiently, it was a great collaboration between the learnings from the small cell and lung group here, having optimized antibodies for small cell subclassification and just porting them over for this project. So again, what I'd like to emphasize here is across this panel of 16 patients, each of whom has multiple different metastatic samples annotated, we see these groups of the ASCL1 subgroup, the amphicrine subgroup, and then a poorly differentiated carcinoma group that's synaptophysin and negative, despite the fact that these patients have late-stage disease with some neuroendocrine features. Another theme that's emerging from this is the heterogeneity within a single patient across the different metastatic sites. So the color code here, blue is for bone and green is for soft tissue. And I'll just call your attention, for example, to this one, patient 7. So these two bone metastases express high levels of AR and NKX 3.1, as well as the cytokeratin marker CK8. So this is an adenocarcinoma, in the usual sense, but they also express DLL3, which is a marker of ASCL1 positive, neuroendocrine cancer in lung, small cell lung, as well as what we're showing here in prostate. And the other two patients make a similar point. So, again, it's going beyond, it's early and maybe going beyond the data a little bit, but the idea that the microenvironment will influence the differentiation state of the tumor cell is a really compelling idea that we're fascinated by. And here's some further confirmation of what I just told you, but now showing you the actual immunohistochemistry in which the rib lesion in this patient is positive for a canonical adenocarcinoma cytokeratin marker, while also expressing DLL3, which is also present in the liver. So I want to now, in the last section, pivot to an organoid culture model in which we want to take these findings and now ask, in a functional, mechanistic way, in greater detail, what are the steps? So this is the work of Wouter, who used his expertise in organoid culture to start a culture from a mouse that's floxed for both RB and P53, looks normal, and then add Cree exogenously to release, the tumor suppressor programs, the programs that are activated upon deletion of these two tumor suppressors. And without really doing anything other than adding Cree virus, you can see what happens if you just look at these under light microscopy. The normal appearance of these cells with round, hollow lumens starts to, get all mixed up. The basal and luminal cells lose their polarity, the lumens fill in, and then after a couple more weeks, you start to see cells migrating throughout the culture. He's generated some great movies of how this migration happens. But importantly, even if you carry this out much longer, you don't get all the way to the neuroendocrine histologies that I showed you in the GEM model. However, if you take these cells and having deleted ex vivo, now put them back into the mouse, and again, we take advantage of this GFP marker, which is activated by the Curie driver, so we can distinguish the implanted cells from the host cells. You see the green, these cells here, you see them expanding. They express the androgen receptor. They do not express synaptophysin. But if you get to this stage on the left and then castrate the mice, give them enzalutamide, and then harvest a couple weeks later, you see that you've greatly accelerated the infiltration phenotype. So this is entirely consistent with what I mentioned briefly from David's earlier paper, that you can accelerate this lineage plasticity with castration therapy. And now we lose the androgen receptor expression and we gain synaptophysin. So again, using the power of single cell analysis, we've done a time course across this ex vivo organoid progression. Then, as I'll show you, also right after we put them back into the animal. And there's, it's an incredibly rich data set, but I want to just make a couple of points on this slide. First, to orient you, this is a time course of that 8-week time course just in vitro at this point. And the way the plots are oriented is in gray here is the start point, and then at the far end is the 8-week time point. So what you can see, and in the one that I'm showing you, is that STAT1 pathway, STAT1 expression and STAT1 activation occurs with very quickly, shortly after the onset of the tumor, after the tumor suppressor gene deletion, and in parallel with that, the activation of a number of interferon response genes, 2 of which are listed here. We also see other programs such as EMT programs, and we also see this loss of luminal features and gain of basal features, sort of mixing of these 2 differentiation programs. So to further, we were very intrigued by the JAK-STAT interferon signal, which I also touched on briefly, sort of jumped out at us when we were profiling the GEM models. So you can quantify that by a score, which is at this low level, in a wild-type organoid prior to deletion of the tumor suppressors, and you can see how it gets activated. It stays activated throughout the period of in vitro culture, and that led us to ask a functional question: is this pathway in any way involved in the plasticity phenotype? So we took advantage of the fact that there are a number of pharmacologic inhibitors that can be brought to bear against JAK1 and JAK2, specifically a clinically approved drug, ruxolitinib. And what I want you to take away from this slide is that, at the start of, prior to adding drug, these cells, this organoid culture has RB and P53 deleted for many weeks and has already progressed to this hyperplastic phenotype where the lumens are filled in. What I hope you can see is that a lot of these lumens have now been restored, but they're still growing quite well. So we're really excited about the fact that maybe JAK kinase activity is involved in getting this plasticity phenotype to unfold. The confirmation that I can tell you about now is that we've used genetics, CRISPR deletion of JAK1 and JAK2. Deletion of either one alone, gives close to this, but the combination looks even better. So we're confident in this model that this has an important role. As I mentioned, we see this in the activation of this same pathway in the adenocarcinoma stage in the GEM model, and that's shown here. It's also present in the EMT cluster, but it goes way down in the neuroendocrine cells that eventually emerge. So I told you that Wouter worked out a way in which he can orthotopically transfer cells that have started the plasticity phenotype ex vivo back into the mouse. And I showed you that after castration, you could see the neuroendocrine cells start to appear. Well, now we've got single cell confirmation of that neuroendocrine program appearing after orthotopic transfer, but this is in an intact mouse. So we haven't even given the extra boost of castration to make it, sort of show up in a more obvious way by immunohistochemistry. And here's a pseudotime analysis, which again this is early stages. We need multiple more time points along here. But what's intriguing to us, and this is the work of Joe Chan and Donna Pear's group, is this bridge. So it's pointing us again to a hypothesis about a cell of origin in this population that we think gives rise to the ultimate neuroendocrine phenotype. So I want to close by saying, which I've already said multiple times, that there's incredible parallels between what we're seeing in prostate cancer and what the lung cancer community is seeing, not just in small cell lung cancer, but also in EGFR mutant lung cancers that can progress rarely to a small cell type, relapse in response to EGFR inhibition. And I'm just, amazed by the number of common transcription factors that are popping up in each of our independent analyses. This, this cartoon comes from a Nature Reviews article that Charlie Ruden's group here at MSK initiated, and were kind enough to invite us in to provide the prostate cancer angle. So I'll close with this summary. It's very clear that these two tumor suppressor genes, and PTEN as well, can cause castration resistance, and the mechanism is through this loss of androgen dependence due to an identity change. I'm excited by the fact that we have models both in mice and in organoid culture where we think we can faithfully get this transition to unfold sort of in front of our eyes so that we can really interrogate in great detail. And as I showed you from the collaboration with the Michael Hafner and colleagues at the Hutch, we think what we're seeing in the mouse is really highly relevant to what you can see in human. The third point is the really interesting role of the tumor microenvironment in sort of affecting different steps of this plasticity. I showed you the importance of that to get to that final stage of NEPC in the organoid orthotopic experiment. But I also hinted at this in the difference in histology between a bone met versus a soft tissue met seen in the human samples. And finally, we're excited by the results we're seeing with the perturbation of JAK-STAT signaling at the early stages, and their obvious clinical implications down the line for combination therapy. So my last slide is just to show you an acknowledgement with pictures of the various people who I've mentioned along the way, and I'm happy to stop now and take questions. Thank you.
Thank you, Charles. Maybe I'll ask the first couple of questions and then go to our ranked list of questions. So, Charles, you mentioned in the normal prostate the different mesenchymal subpopulations. I'm wondering if you've had a chance to analyze stromal populations in the GEM tumor models. Do the tumors change the nature of those populations, and can you speculate on what signaling might be occurring between those populations and the tumor cells in the GEMs?
The answer is yes. We are analyzing those populations. They're much earlier in terms of sophistication of how well we appreciate the changes, but there absolutely are changes for sure. An example in your model is that one of the mesenchymal populations seems to drop out, whereas another one expands. And there are, so that's a phenomenon at this point. But we know the, growth factor profiles of these different mesenchymal populations. So it forms a hypothesis that we can test. And I guarantee you there are ligand receptor pairs to interrogate there that will be of great interest. But we're really too early on to, to say anything beyond that.
Okay, let me go to the question of A panel. So the highest ranked question is, do you see spatial heterogeneity within a single tumor? For example, do certain cell types physically cluster together, or are the cell types evenly distributed throughout the tumor?
I wish I could, pass the ball to Michael Hafner on that one, but I know for sure that the neuroendocrine clusters cells sort of start to burst as almost like, a cluster of grapes or something within an adenocarcinoma background. They're not single cells sort of dispersed uniformly. So it sounds maybe a little teleologic, but I get the sense that there's some signal that gets them going from a microenvironmental probably juxtaposition, and then they can take off. There's a lot more behind to go to help answer that question more fully, but I don't think I have the pathology skills to do it correctly.
Okay, the next question is, do you see any differences regarding the immune cell infiltration among the different subgroups?
Yes, although again, like the stromal question, we're a little early on that. I can tell you that in the, again, the TKO GEM model, there's a very large tumor-associated macrophage population that appears quite early. I say tumor-associated macrophages because they're not there in the normal at nearly that level, and they have gene expression profiles that match signatures that you can find in the literature. What are they doing? Not clear yet, but obviously a very tractable question to address through a whole lot of work from other labs in which you can deplete macrophages. I'm specifically thinking of Jeff Pollard's group in breast cancer, where they've been shown to be involved in metastasis. And there's some really great genetic reagents to do that. So that's high on the list. Whether that's unique to that model or more general, again, too early to say, but we have some data in a different model that's a pure adenocarcinoma model, which shows these macrophages there as well. So I don't think it's just a neuroendocrine transdifferentiation association.
Perhaps an inflammatory sort of response?
Yeah, sure, but yeah, which means it's, is it cause or effect or cause or whatever.
Right,
but
right.
But there is a fair bit of compelling data in other tumor types that you mess with those macrophages and the tumor doesn't grow as well.
No. Okay, the next question is a long one. So in your recent cancer cell study 2020, you highlight 4 transcription factors that contribute to the activation of a non-luminal lineage. Included in this was NR3C1. Considering your prior studies on glucocorticoid receptor taking the place of AR, is your current model now that GR can replace both AR and create a new AR in different lineage, or an AR in different lineage through plasticity? And what defines which GR action would dominate?
Wow, that's a great question. I hope the originator of the question will reach out to me separately, because I'm honored that you're reading our papers so carefully. So this is GR itself, which years ago we published could replace the androgen receptor and maintain a sort of luminal adenocarcinoma phenotype. It comes up in the paper that the questioner is referring to in the context of deletion of CHD1, which is a chromodomain protein that many labs have worked on in prostate cancer. It came out as a hit in a screen for enzalutamide resistance in vivo. So in the context of loss of that chromodomain and then changes in chromatin that are a downstream consequence, I would say it looks like GR can now do things that are different than it does in a pure normal adenocarcinoma background.
Okay, thank you. The next question. So heterogeneity is observed at various levels, genetic, epigenetic, transcriptional. In your study, except for the deletion of RBP10 and P53, would you expect other factors that can drive this heterogeneity or plasticity? If yes, can you speculate on what regulates that heterogeneity and plasticity?
Well, I certainly think other factors can, contribute. I'm not going to, say these are the only 3, because I'll be proven wrong tomorrow. So, these are the 3 that really jump off the page when you look at the frequency of their alterations in prostate cancer. But I'm sure that more will come. there's a second part to the question, which I had an answer to, but I can't remember.
Yeah, the regulation of that heterogeneity. I think we've already touched on that in a couple of questions.
Okay.
The stromal environment and so forth. So I'll just add one little thing to those three genes. If you look in the TCGA pan metastatic cancer analysis, those are the three top pathways in metastatic cancer. So it could be that those three are the three most important, but I agree with you. There certainly must be others. So we have time for maybe a couple more. Here's a quick question. Do you think CDKN2 inactivation or cyclin D amplification can mimic the effects of RB inactivation in lineage plasticity?
I'd almost want to ask you to answer that question, David, because that's your field.
I would say no, based on data not from prostate cancer, but from lung cancer. So in lung cancer, the adenocarcinoma to neuroendocrine transition also occurs in the context of molecularly targeted therapy, in this case, EGFR TKI therapy. And RB loss and P53 loss have been associated with that lineage plasticity, but P16 loss occurs more frequently, but has not been associated with lineage plasticity. But this is very early in the game, so it's still possible. But so far, it looks like RB has some unique role.
The only thing I would add is that, I mean, this is not what the question was about, but CDK4 inhibition in breast cancer is obviously a game changer. And I feel like, we have completely ignored it in prostate cancer. It's not what the person is asking, but it's like shame on us for not looking at that more directly.
Yep. It may be a differentiation agent in some way, yeah. Maybe we have time for one more question. Does the heterogeneity that you observe correlate with any type of treatment, like a guide RNA or drug agonists or anti-androgens, the duration of treatment or type or metastasis, or do you think it's due to differential mutation? So I guess the question has to do with whether the heterogeneity you see is genetically based or epigenetically based or therapy based. I think that's the question.
Okay, well, yeah, maybe what else? I can give some factolets that might help, but so the first point is that the plasticity happens really fast when you give the anti-androgen therapy. So, it's reversible at a certain level. I don't want to, say completely. It is definitely epigenetic, I think, based on what I just said. I think the heterogeneity that we see, I think you induce a program that, let's just call it plasticity. I mean, we'd love to have a molecular definition of that, but that's almost like the start of an evolutionary experiment where various pressures then, maybe it's spatial as in that early question, lead to the outgrowth of this pathway versus that pathway, all those different sort of tantalizing trajectories from that central phenograph that I showed you.
All right, maybe one more question on a topic that we haven't covered, and it has to do with metabolism. Have you studied metabolic flux in your models, especially given that it has been shown that the plasticity and energy metabolism supports the divergent demands of lineage specification and can mediate crosstalk between signaling, metabolism, and epigenetic regulation? And I guess in particular in relevance to the role you're highlighting for JAK-STAT in potentially metabolic homeostasis.
Yeah, it's a great question. We're not looking at it yet. I have lots of colleagues here who are experts in this, so you just gave me some great food for thought to go down the hall and ask.
All right, thank you, Charles. I think we're at the top of the hour, so we should conclude. And I'd like to thank everybody who attended and participated.